Welcome everybody to today's Phenomics webinars. As many of you may know already, the Phenomics webinars is a webinar series on phenotyping and phenomics topics organized by the IPPN, our international network of plant phenotyping institutions and companies, the Emphasis Network, the European Plant Phenotyping Network, so to say, and the Infrastructure Access Project EPPN 2020. And although EPPN 2020 has reached the end of its project runtime, it provided us, among other things, with the opportunity to listen to very interesting talk. And one of these talks that is titled today is the importance of integrating high-throughput phenotyping with anatomical traits of leaves to help understanding plants' acclimation to changing environments. So this talk will be given by Dr. Chiara Amitrano from the University of Naples, Federico II in Italy. Welcome, Chiara. Good to have you here.
Thank you, Philippe, and hi, everyone. I know it's a very long title. Sorry about that.
Oh, don't mind. It's just me that is somehow unable to read it at full length. Before we head into your topic, Chiara, we would like to get to know you a little bit better. And in order to do this, I read, of course, your biography. And there I read a lot of interesting things. So you graduated in 2016 at the University of Naples. And then in your thesis, you specialized on space ecology.
Yeah.
Among, of course, plant ecophysiology, but we'll get back to this. I found this highly interesting, of course. In July 2021, you got your PhD in sustainable agricultural and forestry systems and food security, also from the University of Naples, in the Department of Agricultural Science. So there you worked with Professor Veronica de Micano and Professor Stefania de Pascal. So the topic of your PhD then was characterization of plant water flows in controlled environment, plant smart sensors. So what does that mean? Controlled environment, plant smart sensors.
Yeah, you will see some of that today because these phenotyping experiments were actually part of my PhD thesis. So right. I won't spoiler you because you will see later.
Yes, yeah, definitely. So how does this go together with space ecology? Also, yeah, space ecology.
Yeah, in my department, but also the Department of Biology, where I graduated from, we have this huge research line about space and the possibility of growing plants in space environment. So I won't talk about it today, but actually I will mention it because all of my studies for my PhD were held in controlled environment. And so for growing plants in space, but also in extreme environments or also in urban context, you need controlled environment agriculture. So that's why these two kind of research are actually connected.
Yes, I see. Of course, controlled environment agriculture has many implications across a large range of applications. I think also this controlled environment agriculture brought you to Arizona, where you had a stay abroad for around 9 months, as I read, at the lab of Murad Kasira.
Yeah, I stayed there during my period abroad, and I worked in a actually controlled environment in a vertical farm. And it was an awesome experience.
Yes, as Murat did present during the Phenome 2020 meeting in one of IPPN's sessions. So thereby I had the pleasure of getting to know him, and he's amazing and incredibly also friendly guy. Very, I can imagine working with him is quite nice. So after you stay in Arizona, USA, you applied for a EPPN 2020 fund to access high throughput phenotyping infrastructure at the IPK at Thomas Altman and Astrid Junker's lab.
Yeah, we wanted to complete our experiments of the PhD utilizing some phenotyping techniques. And so we applied for the EPPM because we didn't have the possibility to do that over here. And actually our project was chosen as one of the winners. So I went there, I stayed there for about 3 months performing phenotyping. And it was my first time. So I learned a lot, and it was actually great to stay with them.
Yes, also Thomas and Astrid are, of course, IPPN members and very active in the community. Thereby, I also know them quite well. So I'm very, very excited to hear about your talk today, the importance of integrating high-throughput phenotyping with anatomical traits of leaves to help understanding plants' acclimation to changing environment. Chiara, it's a pleasure, and the stage is yours.
Thank you, Philippe. I will share my presentation. And sorry again for the length of the title of my presentation. All right, can you see it? It's all working. Can you give me a feedback if you see the screen?
Yes, everything is visible and everything is good.
Okay, thank you so much. So, thank you, Philippe, again, and welcome everybody to these Phenomic webinars. So I'd like to start this presentation actually showing you our department. This is the Department of Agricultural Science of the University of Naples, Federico II, where as you already know, I completed my PhD in July of last year, and I'm actually a postdoc. So this is a beautiful royal palace, which is in Portici, a small town near Naples, and it was built by King Charles of the Bourbon family. So it's actually a old building, but it's very, very nice working over here every day. So at the rooftop, there is actually our lab, which is the plant and wood anatomy lab. And the heads of lab are Professor D'Amico and Professor Di Pasquale. They are both botanists and archaeobotanists. So what we do basically is study plant and wood anatomy, as the name said. But actually, we are a multidisciplinary lab with where many people with different backgrounds. And here I try to summarize some of the most important topics of our lab. So we studied the adaptive strategies in Mediterranean species, the xylogenesis of terrain in response to environmental changes, the morpho-anatomical plasticity linked to efficiency in different plant species, and I will talk about it today, historical biogeography of good species, the interaction between man and forest throughout the Holocene, and the adaptation of plants to extreme environments, as I said before, also connected to space research. So basically, I highlighted these two topics because they are the one I mostly care about, and I did research for during the last 4 or 5 years, maybe. So I'm also collaborating with other researcher and professor in and out our department. Our approach is almost always the same. We try to set up a multidisciplinary study going from small scale to a bigger scale. So in any study we do, for example, this one refers to some research on vineyard we have going on right now. We try to study from the particular, so anatomical studies of tissue, like for example, roots and stem and leaves, to correlate these studies with the physiology of plants. So for example, here shows some gas exchange measurements, to the quality of fruits and leaves, and also to a bigger scale in the sense of proximal sensing, for example, as I did, and as I'll show you today for the phenotyping study, and also collecting some data from satellite and so on. So working in a lab which mostly work on plant and wood anatomy, the question we received so often is this one: is plant anatomy worth studying, and why? So of course, our answer can only be it's very important and it's definitely worth studying, because we actually believe that plant anatomy, it stays in the middle between the cell biology and the plant morphology. And also it reveals the connection between the structure to function. So we focus a lot on the structure to function relationships of these many species in very different environments. So of course, the context of this research, and actually I believe of any research concerning plants nowadays, is basically on climate change. I also believe that anyone connected here knows the terrible impact of climate change on agriculture. So just to mention a few, the increase in atmosphere CO2 concentration will determine, and is actually determining right now, a rise in air temperature. This consequently affects plant evapotranspiration, enhancing plant evapotranspiration, which is impacting crop production all over the world, but especially in arid and semi-arid regions of the world. Plus, global warming will also exacerbate problem already existing right now, leading to important changes in the atmospheric water vapor, which I will speak a lot today, and also on it will change the pattern on precipitation amount. So basically, the research I conducted for my PhD, which also comprehend this phenotyping experiment, it was held in a control environment. So in control environment, I had the possibility to change the internal parameters, so temperature, relative humidity, to simulate some stress connected, for example, to climatic changes. And this had two main points: to understand how plants will react to this sudden stress and these changes in environmental parameter, to optimize the production of edible plants on Earth, and also, as I said before, the production in extreme environment. So working in controlled environment agriculture was actually very, very helpful. So in a closed system, this is what happened. So all the environmental factors, as temperature, light, relative humidity, will affect plant morphology, but also they will change, as I show you later, the internal organization of plant tissue, so plant anatomy. This, of course, will change the physiology of plants in terms, for example, of transpiration, gas exchange, and in closed system, it will change back the environment. So it's very important that to show this as a loop, and that all these parameters should be controlled and modulated together. So for my research, I focus a lot on this parameter, which is the vapor pressure deficit, or VPD, which basically drives all the water fluxes in plants. So for example, plants get water from the roots, in the soil plant atmosphere continues, it goes through the stem, then to the leaves, and then it will go back to the environment. So the VPD level of the environment, it's very, very important to control. And of course, as you can see over here, leaf anatomy has a central role in the evaporative process, but not only. It actually, we will discover through this presentation, its central role in regulating a lot of process. So here there is an example which I call the productivity versus survivability trade-off. So at low VPD, which is considered in our experiments like an optimal condition, but it's not only that. So it means low temperature and high relative humidity. The atmosphere is full with water. So what happened is that plants experience low transpiration. The stomata, which are a very important part of leaf anatomy, are open. So there is an enhanced conductance, so there is a good amount of gas exchange. However, plants in a humid environment may be more susceptible, more vulnerable to diseases. Instead, at high VPD, so this means high air temperature and low relative humidity, the atmosphere, as we can see over here, is very dry. So basically what happened is that the transpiration of plants is high. So plants try to enhance the safe mechanism. So they close their stomata. And of course, this will reduce the conductance and will reduce the photosynthetic amount. But of course, on the other hand, the plants may, if the VPD is too high and the transpiration consequently is too high, the plants may dry and wilt. So basically what happened in the field where there are some environmental stressors due to climatic change is that like the water and thermal stress is basically this showed over here on the right. So there are some loss of yield and biomass. And of course, if we're talking about food plants, there will be also some loss of income. But why this happen and how plant can survive this? There are plants which actually can adapt to live in extreme environments like in deserts, for example, or in water. So why did that happen? Because these plants already have some morpho-anatomical adaptation, which allow them to live in this different environment. And I put some picture over here, for example, a needle-like leaf or an aquatic plant or a leaf which has developed a very cuticular thickening. So these are plants which are, as I said before, already adapted to live in extreme environments, so already adapted to live in subjected to these environmental factors. However, it can also happen that plants, the same plants of the same species, if it's in two different environmental conditions, put in two different environmental conditions, can change completely its morphology and its anatomy to adapt to the new condition. And this is very, a really nice example is that of sun leaves and shade leaves that almost everybody knows. So plants, these actually sometimes happen in plants, but it can also happen just in a few leaves of a plant. So sun and shade leaves develop a very different morphology, usually, but it's also depending on the species. Sun leaves have very smaller and thicker leaves compared to shade, and this probably happened because shade leaves, they need to capture more light, so they try to enhance their area in order to capture more light. The internal anatomy of the mesophyll is completely different, usually, but again, it depends on the species. Sun leaves presented 2 or more layers of palisade cell and a different distribution of the chloroplasts on the palisade cell compared to the shade leaves. And the pigmentation, it's another strong different characteristics between sun and shade leaves. So sun leaves appear more lighter, a green which is lighter compared to shade leaves, and this is of course due to the chlorophyll content. So shade leaves, for the same reason, to capture more light, they develop more chlorophyll, and so they appear darker. Okay, so it goes without saying that, anatomy drives the acclimation. In the same way, this is a stupid example, but actually, if you think about it, in the same way of humans, as these two people, an athlete and a man who have gained too much fat, they cannot have the same total lung capacity. In the same way, plant anatomy established the limits of its functioning. So these two plants meaning that the first one, of course, this is just an example, but it's flowering and it's good and flowering. And the second plant, which is suffering from, for example, some water stress or some disease, they cannot have the same photosynthetic capacity. So once established the importance of tissue anatomy, there are still some open issues in the literature we try to respond to during these experiments I will show you soon after. So the first one is whether and how changes in leaf morpho-anatomical traits can drive the photosynthetic acclimation to environmental conditions. So bearing in mind that in the same species, different environmental factors can lead to different structure. How can this internal structure drive the acclimation to the physiological acclimation? So this is an open issue or a controversial point. How plants react when there are some changes in the environment, which does not mean climate change, but also short-term environmental change, and how this is connected to their morphological plasticity. This is another open issue. And also how the interaction between the VPD and other environmental stressors, so for example, I will show you some experiments with water stress, will affect the morphophysiological development. And also we're working on how in reduced volumes, of course, controlled volumes, that we use for urban application, but also for space research, the VPD level may affect the plant development and the whole water flows. And I will talk a little bit less about it because otherwise it would be a never-ending presentation today. So during my PhD, we started from experiments in small growth chambers like this one over here, and working mostly to unravel these open issues. And we set up experiments growing plants at different VPD. We usually had this experimental design where we grew plants, edible plants, mostly lettuces, under low VPD, which was considered optimal, let's say, environment, and under high VPD, which was considered a suboptimal environment. And so we try to understand this point. So how changes in anatomical and physiological traits were led by the VPD. We worked a lot on the short-term acclimation to changing climate. And we also try to predict and modelize the behavior of these plants in controlled environments. So I put it over here, some of the paper which we published during the PhD time. And these are actually some results from these experiments that confirm that plants developed under low and high VPD, they develop a completely different morpho-anatomical structure. For example, plants under low VPD developed more stomata, so a higher stomata density, and stomata were also smaller compared to plants at high VPD. They also developed more veins and smaller veins, and a completely different structure of the mesophyll. So, for example, plants grow in under high VPD, so in the one which is considered a suboptimal environment. As you can see from this picture on the right, they develop more intercellular spaces and a palisade parenchyma, which was smaller. And what does this mean? Actually, recent research, and we kind of confirmed that, show that plants with more intercellular spaces will be more resistant to water and also to gas flow. And this is how it will appear very, very simplified, because the pathway through the intercellular spaces will be longer, and so it will increase the resistance. Okay, so with this experiment published here in EEB on Vigna radiata, which is monthly, we tried for the first time this short-term acclimation of plants. What we did was to grow monthly plants in two different growth chambers, so under low VPD and again under high VPD. After 30 days, we performed all the analysis, meaning both anatomical and physiological analysis in terms of gas exchange and chlorophyll fluorescence. We also have extracted some pigments like chlorophyll and so on. After these 30 days of growth, where the plants were have very developed leaves that cannot change anymore their organization of their tissue, we decided to subject them to the short-term changes in the environment. So we basically transferred half from the plants to the opposite growing chambers and repeated all the analysis. So what happened was that the VPD levels determined a different leaf anatomical structure, as we actually expected. And also we witnessed a different coordination of these anatomical traits. For example, this is a graph showing the coordination between stomata and vein densities. And these actually drive the acclimation after the transferring. So for example, here you can see a table regarding the net photosynthesis, and we can see that what we call HV plants, so plants grew at high VPD before and after the transferring to a better environmental condition. They had a less efficient, let's say, anatomical structure and could not acclimate to the best conditions. So they did not change their photosynthesis before compared to before the transfer. So this was a first proof on how the anatomy can lead to physiological plasticity, also when they are subjected to short-term changes in the environment. So it's all mediated by leaf anatomy. So as I said before, at this point, we decide to apply, and then we won a grant in the EPPN transnational access to perform this phenotyping experiment at the IPK in Gattersleben. So as I said before, there I worked with Thomas Altman and Astrid Juncker, and we won a grant with this project, which we call automatic phenotyping platform to improve lettuce water use efficiency under different VPD and watering regimes. And we were allowed to use the small plant facilities that we had over there, which is this one showed in pictures, which actually is not small at all. It's a working chamber, and we were able to accommodate about 1,000 plants. And we try to maintain the same or similar experimental design of the previous experiment. So we started choosing 2 VPD levels, in the low VPD in a chamber and the high VPD in another chamber. And these were chosen considering basing on previous experiment. And then we worked on green and red Salanova plants, as we did before, because we knew well this kind of plants. But this time we added another stressor. So half of the plants were well watered, and for well watered meaning 100 field capacity, whereas half of the plants were drought stressed. And this was a drought stress of about 50% of the complete of the field capacity. And already from here, you can see how different they look. And the same we did in the high VPD condition. And again, you can also see, because these are all pictures of the same day, so you can also see how plants develop very differently. So after that, after 12 days of growth, we did not transfer the plant from a room to another, because of course phenotyping is all automated, but we changed the environmental condition in the chamber. So we passed from low to high VPD to the opposite environmental condition. And we use RGB, fluorescence, infrared cameras every day. And some days before and some days after the switch in environmental condition, we also perform some chlorophyll fluorescence. So here is just a brief video to show you the chamber. Basically, here in this room, there were the cameras, and I put here some images, and it was all automatized. We used this blue rubber mat to eliminate the evaporation from the soil. So, and also for pictures, it's very used in control facilities of this time. And it was all automatized. So the watering station, it was already set up to water some plants at 100% field capacity and other plants at 50% field capacity. So basically everything worked well. And now I will show you some results. The first results are the growth, basically. This first graph is about actually it came from RGB cameras, and it's about the plant area. And what we saw was very interesting. So the higher plant area was, of course, under low VPD, as we expected, because, as I said before, we consider it to be an optimal condition. And this basically were plants subjected to what we call well-watered plants. But here something happened. So plants at high VPD, but the well-watered plants under high VPD grew less, but the red one managed to reach the same area. So there are no statistical differences of those under low VPD. And however, the difference is very slow, very differently, considering the low water regimes. Here, there is a huge difference between plants at low VPD and plants at high VPD. So this struck us the most, especially the behavior of these red plants, but it was actually probably cultivar related, and we will see later also anatomical related. So the same happened basically with the weight. So here I have highlighted the fresh, but also the dry weight, which were higher, of course, under low VPD and in well-watered plants, as we expected, and it was visible from the picture. Because I forgot to say that after the phenotyping, we performed manually the fresh and the dry weight, and we brought back at the Plantations with the anatomy lab some samples preserved in a fixative to perform all the anatomical analysis. So these are some pictures from the anatomical traits or light microscopy pictures. And as we can see, the water stress, but also the high VPD conditions, they did not affect the overall organization of the leaf tissue in qualitative terms. So if you look at these pictures, you cannot see a plant which is suffering. However, there was a different organization of the tissue, and this is evident for example, in the difference between the well-watered plants and the low-watered plants, which developed a thicker lamina, for example. Plus, it was very, very evident in the veins and in the stomata organization. Plants under low VPD developed more veins, but especially the one at low watering level. So probably this was an acclimation mechanism to bring more water when they had basically less compared to the well-watered plants. And also at low VPD, it's very visible in this picture, they developed more stomata, but this time the more stomata were where the water was good. So in low VPD, well-watered plants. And this organization influenced a lot the photosynthetic machinery and the capacity in acclimation. So for example, this is just a graph which refers to the FVFM index. This was calculated with the chlorophyll fluorescence analysis imaging. And it's worth noting that switching the condition from low VPD to high VPD, there was, of course, a reduction of this parameter, and vice versa. So switching the condition from high VPD to low VPD, there was actually a slow enhancement, but it was still significant in this plant. However, it was also interesting to notice, and it was not the first time that happened in that facilities, that all of the plants which may have been particularly resistant were above the 0.8, which is often considered a threshold for stressed plants. So also plants grew at high VPD and also that with low water, they managed to have this FVFM value, which was above the 0.8. Whereas over here, we will have a panoramic view of just two of the traits that are selected. These are the yellow to green and the lab B traits that are both considered proxies for, let's say, stress detection symptoms, stress levels. And they showed that the early stress symptoms were actually detectable thanks to the phenotyping. And they were, as expected, more evident in H plants. So plants grew at high VPD from the beginning, and also in low watering plants. So at this point, what we did was try to, let's say, combine some anatomical and some physiological data. And here I will show you. Okay. We ran some PCA. The first four I will show, there will be all PCA run with the anatomical parameters. So for example, the stomata area, the palisade thickness, the thickness of the spongy parenchyma and so on, the vein density and so on. And here from the first graph, and as I was saying, they are all run with the same metrics, but they are highlighted different things. For example, the first graph, it highlights and separate the data between green and red lectuces. So basically, they here are divided for cultivar. And we see there is not a really division of this data. They don't split for cultivar. They are all mixed up together. They started separating when we highlight data for low water and well-watered plants. And they actually are more and more separated for VPD conditions, so low and high VPD, and all the conditions together. So we can say that VPD had a very impact on these anatomical data. So they developed, as I showed you before, a different anatomical structure. Whereas these are the four PCA in the same, divided in the same way of those before, but these actually are with all the, not all, but a lot of phenotypic indexes. And here we can see a very different story. So the first graph is actually very explicative because the data are very, very nicely divided for color, so for cultivar. And what does this mean? This means that these cameras could easily detect the stress already in progress in plants. They can divide the plants for, let's say, for color, but they actually were not able to detect differences in their structural organization, which was actually responsible for the greater or for the fewer capacity in acclimation when we change the environment. After this, we also tried to perform some correlation between all the traits we measured, so both anatomical and also phenotyping traits. Of course, I won't show you all of them because they are very, a lot of graphs, very complicated to read. But for example, I have highlighted this one. As you remember, the well-watered bread plants under high VPD, they managed to reach the same values of plant areas of the plants under low VPD, again, well-watered. And so I have highlighted this one difference, which is a correlation, a positive correlation between the stomata density and the fresh weight. This probably explains how the improvements in leaf area, because we know that higher stomatal density is also known to improve the carbon gain, also to maintain a certain kind of homeostasis in plants between the carbon gain and water loss. So this could be an explanation on the phenomena we experienced before. So I will just run to conclusion. And in conclusion, we can say from the studies that the VPD and in general the environmental factors in the same species change the morpho-anatomical composition of tissue. And this is a fact. And sometimes it's species specific, or even as we said here with these two cultivar experiments, sometimes it's also cultivar specific. Also, high throughput phenotyping, it's a very good instrument. It's a powerful tool for the early diagnosis of stress symptoms in these plants, like differences between green and red Salanova, or differences between well-watered and low-watered plant. It was able to detect the stress very soon during the cultivation. However, we actually believe that integrating this study with also the study of leaf anatomical traits will allow us to understand the mechanism in acclimation and how plants can, and how and if plants can adapt to react to this changing environmental condition. So we think that the combination of the two techniques, it's very important to close the loop. And these data are actually from the phenotyping experiment are actually under review in Planta Springer. So let's hope that this paper will go through without problem. And also I'd like to thank you, the EPPN, for inviting me today here, and all of you for listening. And now if you have any question, I'm here to answer.
Thank you very much, Chiara. A very interesting study, very comprehensive study, also of plants' reaction to variations in water availability and water pressure deficit. Of course, in nature, I mean, most of the times these two factors even co-occur. So whenever you have at least a dry, hot climate, you have soil, water scarcity and high VPDs. So this is a very relevant storyline that you showed up here, that you put up here, stressing the need to investigate this topic further. Thank you very much for this highly interesting talk, and I would like to open the discussion.
Sure.
So first question comes from Alex Burgess, and he would like to ask, how old are the plants? Were you targeting developmental, acclimation, or dynamic? Depending on your answer, what do you think you would see if you target a different plant age when growth was fixed or still being plastic?
Okay, this is a very interesting question, because I actually was thinking about it before starting these experiments. So what was best to do? So if targeting a developmental stage, or if to explore these where plants were still growing. So basically what we did in these experiments was to analyze the anatomical structure of the leaves which were already formed. So there were no young leaves, they were the most expanded one with an anatomical structure which was already formed. So when we switched the condition, we said we were able to detect if a plant in that condition, so to simulate if a plant in already developed was actually still able to have some plasticity and acclimate to the new environmental condition. And sometimes we saw that it could not, and sometimes we saw that it could. So this was very dependent on the study we performed. But actually, I'd like also to try the same setup, but to repeat the analysis during the growth to see the differences between very young leaves and older leaves, let's say.
Yeah, how old were the plants when you applied the treatment?
They were like 20 days old, and of course, we chose the leaves which were all in the same developmental stage, expanded and formed. In the phenotyping facilities, we had to cut the experiment first because we were using the small plant phenotyping chamber. They actually use that for Arabidopsis. Instead, our lettuces, especially the well-watered one, grew faster and faster. So as you see from the pictures, they actually had covered the whole pot. And so the cameras, after a while, could not detect the whole plants anymore. So we had to stop that some days before.
Yes, unfortunately, one of the limitations of high-throughput plant phenotyping systems is that they can be quite picky for the size of the plant, for which they are thought to provide accurate measurements. So, yeah, that is, of course, a drawback on that. So another question on the system you used at IPK. Also, Alex asks, I haven't seen the IPK setup before. Why does every other station have weighted bags in it? Is that necessary, or can you actually fill every shuttle at full capacity? I think that has been done in order to have some control for the measurement, right? And whether there is a drift from beginning to the end of the shape.
Yeah, actually, it's for both things, the one you just said, but also for the one I was saying before. So since my plants were expected to grow after the pots, we didn't want that put two plants together so that moving, they would cut some of the leaves. So that's why we chose this setup with every other station with the plant and every other with a bag full of sand. And it should be sand or something in order to put weights actually to simulate the plants over there.
Yes. Yeah. A small remark, of course, at IPK, they have two different systems, two different conveyors, one for big plants and small plants. But for each of the system, of course, experiments can only work under the given preconditions. So
I chose the small one. Sorry to interrupt, because it was fully controlled. It was set in a walk-in chamber, whereas the one for big plants, it's actually for very big plants, and it's also in a greenhouse. So I was more interesting in the small one. So I try to adapt their system to my plants, and so we have to compromise.
Yes, but you found a way. I mean, a good one, I think. So next question from Chesham Hassan. Asking, are you planning to understand the molecular mechanisms behind these plant responses to changes?
Yeah, I'd really like to do some molecular measurements. For example, we were very interesting in studying the aquaporins, and we are not able to do it in our lab. So we are actually looking for connection in order to do that, because I believe that it will be really important to complete these studies on water fluxes, VPDs, stomata. So I'd actually like to do that.
Do you fancy any specific kind of measurement system or technology to investigate the aquaporins in the roots?
Yeah, I would like to do everything, but I think I have to talk through with someone who is an expert in that area and understand which one of these methods is best for my research. So.
Yes. So in any case, if someone would have also a question maybe to you, or is open to discuss and collaborate with you, how can they contact you? Is there a website?
Yeah, I put my email in the presentation, so you will find it over there, or we have the page of our lab, the PVWA lab. It's on every social, so it's very easy. And please contact us, because it's very important to have these moments in order to create also connection. I strongly believe that.
Of course, this is also a scope of the IPPN. So for anyone who is still asking how to contact Chiara, feel free to also send a mail to ippn at plant-phenotyping.org, and then I will gladly redirect you. Next question. So in terms of combined observation of physiological and anatomical traits of plants, there are few tools available which allow investigation of both at the same time. This pretty much connects to what you want to do next. However, using hyperspectral and or X-ray CT systems are to some extent making this possible. What is your opinion on this? Are you planning to use tools like these, or do you see other applicable technologies emerging?
Yeah, I think the CT will be very important to do that, because you are able in a certain measure to understand both the anatomical and the physiological part of plants. But I believe that you will still need some in-depth analysis of plant anatomy. So you still have to actually cut and open plants, or in future, who knows, maybe there will be other techniques which will allow us to do it in a non-destructive way, like the phenotyping versus something deeper, in order to see the different composition of tissue. This will be actually awesome to do. And another thing I'd like to do is actually to study the dynamics of stomata opening and closing in vivo, so during the growth of plants. So that will be the goal. We are actually working to reach that.
Okay, in your lab, you mean you are actually currently designing?
I mean the research worldwide, not only in my lab.
Yes, that's true. Next question: do these morpho-anatomical changes have any impact on the nutritional profiling, so of the stoichiometry of the plants?
Yeah, actually, this is a very interesting question, because it had, of course, according to my experience, in some of the paper I showed you before, I have measured also some nutrients are mostly antioxidant and some chlorophyll content, carotenoid content, all with spectrophotometric analysis. And we actually saw a huge difference, but I believe it's mostly connected to the different environmental conditions. So it's the VPD, or the water stress, which drives the development of these nutritional compounds, let's say. Instead, from a morpho-anatomical point of view, we have experienced some changes in the composition, not in the composition, in the distribution of these phenolic compounds, for example. And this is very dependent from the anatomy. So, for example, in some of the plants, you could see the phenolics that were accumulated all below a certain point, or I don't know how to put this. So maybe they were accumulated up, or they were all down. So it's more the distribution connected to the anatomy of the plants, whereas I think it's more the environment to actually change the amount of these nutritional compounds. But since the environment changed the anatomy, we can actually say that it's all connected again.
Yes. So yeah, good. So next question: the results indicate that plants which once adapted to suboptimal conditions on an anatomical basis limit their carbon gain in favor of conserving water, even after they have been brought back into optimal climatic conditions. Have you investigated bringing, how long did you observe the plants, and will they maybe just take more time to adapt again back to optimal conditions?
Okay. Actually, we didn't do that, but in the first experiment I presented, the one on mangbean, we maintained half of the plants under optimal condition, and we brought half of the plants under the suboptimal condition. Then we did not brought back, but we continue monitoring the other plants just to have a very tight control. So it would be interesting to see if they readapt and how long they will take to readapt to their condition in which they were grown. This is a good point. We should definitely do some research into that.
Mm-hmm. And did you also observe different genetic lines of the same species?
No, actually, we did not. We just used for this experiment these two cultivars, which are the green and red Salanova lettuces. And we actually found out that they are very resistant to drought stress, because you see, they reduced their dimensions a lot. And of course, they developed a whole morpho-anatomical traits very different from the well-watered plants, but they actually were good. They stayed good. And also the symptoms were, the early stress symptoms were detectable, but they still were acceptable for plants. I expected a lot worse.
So for the future, Chiara, what are you currently working on, and what are your future plans? Will they pick up the research? Oh my God, today is very hard for me to speak. Will you continue that line of research, and what you're gonna do next?
Yeah, I will definitely continue this line of research, but actually for every experiment we set up, for every line of research we have, we try to maintain the same approach. So I will definitely continue in this way. And also we are working a lot with my group, with Professor D'Amico and Professor De Pascal and all the other collaborating in space research. So right now we are doing some experiments in very small grow chamber, try to optimize all the research allocation for the growth in a completely different setting. And we are using very small plants like microgreens or baby leaf. So that's what I'm doing right now.
Mm-hmm. So will we see you or will you attend any conferences this year? I mean, we are still under the influence of the COVID restrictions, I think, all over the world. However, currently it looks like, at least for the summer period, we are facing some lesser restrictions. So are you planning to attend anywhere across the world? Any conference?
Yeah, sure. I hope to attend some conference in person for a change this time, this year, but we actually don't know. For sure, I'll go to the EGU in Vienna at the end of May, I guess. And then I'm still planning what to do, because it depends on online and travel restrictions. So we will see during the year, but for sure.
Thank you very much. That was it for the questions. I would just quickly hint you maybe at our working group for controlled environment, plant phenotyping, that is also led by Thomas Altmann. PPN working group. You will find them on our website, and I can also bring you in contact. He's also in contact with Murat Kassira or Jerry Stutte, and they are, I think, the right partners if you're looking for collaborations. And of course, during the IPPS, the International Plant Phenotyping Symposium later this year, in the end of September, at the University of Wageningen, they will also be having a workshop on controlled environment agriculture, possibly then with a lot of other researchers from around the world. So I would be very happy to see you there, and this would certainly be a good point of connection.
Yeah, it will be great. I'll try to be there, of course.
Very good to hear. So hopefully we will see here again, the IPPS is from 26th to 30th of September, this time in the beautiful Wageningen. And yeah, I would be actually happy to meet all of you here, and also Chiara, hopefully then with new results on her very exciting research topic. Thank you very much, Chiara. It has been a real pleasure. And the last words are...
Thank you. The pleasure was mine.
Okay, I'm saying goodbye to all. See you next month. Bye-bye.
Perfect. Bye. And thank you all for listening.